Okay, well, it's great to start the new term with a seminar with Bill Newsome. Bill has come from Stanford. He's actually here for a few months on sabbatical, making research, and today, as you can see, he's going to talk about free choice in the brain, interpreting volition in a post-Libet sciences. And he's been doing some fascinating research, and today, as you can see, he's going to talk about free choice in the brain, interpreting volition in a post-Libet era. And I presume you're going to tell who Libet was as well, for those of us who don't know. So, Bill, over to you.
Thank you. It's a pleasure to be back here at St. Edmunds. I was here about 2 years ago, I think, in 2010, and I had a very enjoyable visit. And I'm back here now for several months, and I have a stack of books about this high I want to read about and think about and have time away from the lab to digest, some broader issues about neuroscience and how it relates to society and the rest of academia. Dennis Alexander, who's conveniently in North America at this time, actually asked me to give this talk on this particular topic, the status of volition in a post-Libet era. And you should all be aware ahead of time here that I don't actually do research on Libetian style decision making. I'll give you a little bit about my background and what I do. So what I intend to do, I've always been interested in this work, and I took Dennis at his word literally to do this. And I learned a lot while I was preparing for this talk. And what I intend to do is, as Bob suggested, give all of you some idea of Ben Libet's basic finding that has cast such a long shadow over psychology and philosophy now for 30 years. And I'll sort of give you all an intuitive feel of what that was all about without showing you any of his original data. And then I will skip a couple of decades to some work that's been done in Mark Hallett's lab at the National Institutes of Health. And I'll review that for you because I think there are many, many methodological problems with the general line of Libetian work. And I'll allude to some of those. And I think Hallett has done the best experiments trying to actually measure conscious volition objectively. And I'll show you what he did. I'll actually take you through some of their data and show you why he believes that he's confirmed Libet's basic results. And then I'll tell you why I continue to have reservations about this basic result. And I will propose an experiment that hasn't been done. I've tried to sell some of my colleagues on this experiment who actually have the ability to record EEGs from human subjects. All of my work is in laboratory animals. And I'll put it out there for you guys to think about. And there's some of you, I'm sure, who know more about this topic than I do. And I may learn that my experiment is permanently flawed, or we may decide that it's a good experiment and really ought to be done, in which case I'll go with more vigor after people who really have the capability to do it. And then lastly, we will discuss what's at stake anyway. We'll get around to a more general discussion, what's at stake anyway in this whole line of research. Okay, so I have a background in the neuroscientific study, neurobiological study of decision making. And my work takes place with monkeys who we bring into a laboratory and train to do different kinds of decision-making tasks. And while the monkeys are doing the tasks, we can record the activity of single neurons from different points in their brain, and we can actually trace signals from input to output in a very fine way. And I first started working on this decision-making topic. My background is really in vision, and I first started working on decision-making in about the mid-1990s. And this was actually the first paper that Mike Shadlin, who at the time was a postdoc in my lab, but has since gone on to much grander things. Shadlin and I published this paper in PNAS in 1996 on motion perception, seeing, and deciding. And the simple little task that we had our monkeys do was to look at a TV screen on which random dots are displayed. And these are twinkling random dots that may have a net motion to the right or to the left. And the monkey simply would look at the look at the dots here. And at the end of the motion presentation, he would, if he saw net continually from quasars, so we can monitor his performance very carefully. So in this example, there's net motion to the right, and the monkey moves his eyes to the right at the end of the trial, gets rewarded for his efforts. And you guys can look at one of these stimuli right here. If this works all right, I'll play the movie, and your job is just to tell me in this little movie whether the motion is moving to the right or to the left. Do you see that? And the answer is Right. Was it? You want me to play it again? This is actually a fairly easy one right here. I assure you, the monkeys can do much better. Okay, so I'm gonna have to go back and see if I can play this thing again. There we go. So hopefully you can see that's kind of drifting right, because there's random motion noise in there, right? And we can make this arbitrarily easy or arbitrarily difficult, depending on how much signal and how much noise we dial in there. So we can get very fine control over the monkey's behavior. And we've had some success over the years in identifying the areas of the visual cortex and the exact neurons and circuits that code the direction of motion, so the sensory signals. But Mike and I had some initial successes in actually identifying higher level circuits that seem to be involved in the process of decision formation, not simply recording what's on the retina, but actually participating in what the animal decides. And we then could trace signals. The nice thing about this paradigm, we could trace the signals out to the motor commands to move the eyes to the right or the eyes to the left, so that we could start getting a picture by recording these signals through the entire brain of the circuits that are responsible for decision making in this simple sensory task. Now, a little bit later, Leo Suzu and Greg Corrado, two graduate students in my lab, started looking at decision making from a very different point of view. And this is a decision about value. We call this value-based decision making, representation of value in the parietal cortex. This is a paper published in 2004. And in this thing, the monkeys are also moving their eyes from one target to the another. On every trial, they have two targets presented. They simply choose one target or the other. But now they have no cue to tell them what's the correct one, what's the correct target. The choice is free in some sense, that we're not controlling it or demanding a particular answer. And what the animal is doing, he's looking for rewards that are hidden basically on each of these targets. And of course, we control the schedule by which they become available, and they become available in a stochastic manner with different proportions on the two targets. So the animal senses these proportions by trial and error. Sometimes he makes an eye movement and gets rewarded, and sometimes he doesn't. And he senses them by trial and error. He estimates the likelihood of success on the two colors, and then he starts devising strategies to exploit what he knows about his environment. And of course, being the demonic people we are, we will switch the contingencies at some point. But that just reflects the real world. Any of you who have children know that the world can change on you very quickly, okay? Right? The emotional tone of the world can change very quickly. But the monkeys learn this well, and what we discovered at the end of a long set of experiments is that the same circuits that seem to be involved in decision formation in the perceptual task are also involved in decision formation in this kind of value judgment, economic value, I'm using it here, economic value judgment. And these lines of work are ongoing. I'd say the best work that's been done in the last 10 years is from Shadlin's lab. And I'm probably inordinately proud of my own scientific progeny, but that's my perception at any rate. But when you get to the end of the day, there's some interesting questions that one would like to ask about decision making that are very, very difficult and perhaps impossible to ask in experimental animals. And the main question that would be nice to be able to ask, but is hard to ask, is what are the conscious concomitants that accompany the animal's decision making? So, for example, we can electrically stimulate some parts of the brain that represent rightward motion or leftward motion, and we can cause the animal to change their judgments from rightward motion to leftward motion, but we don't know what the animal's actually seeing. So if the monkey could talk to us, would he say, hey guys, I saw rightward motion on that trial, and I chose the right dot. Why didn't you give me a reward? Or would he say, you know, I saw leftward motion, but I chose the right dot. I don't know why the hell I did that. Right? And both of those subjective interpretations are consistent with the data the objective data that we actually measure. So that illustrates, you know, where in the laboratory, we actually ran up against this roadblock. This isn't just a philosophical issue for us anymore. We actually experimentally run up against a roadblock and real limits to our interpretations of data. So to start asking questions about conscious concomitants of decision making and the role of conscious awareness in decision making, you really have to go to human subjects. And I haven't made that step yet, but this guy here, Benjamin Libet, made it a number of decades ago now, in 1983, with this very famous paper that was published in Brain in 1983. This is Ben Libet, by the way. And the title of the paper was Time of Conscious Intention to Act in Relationship to the Onset of Cerebral Activity. And this onset of cerebral activity is an EEG measurement called the readiness potential. And this paper and the subsequent studies that sprang from it or were inspired by it have cast this long shadow over psychology and philosophy ever since, because the central claim of the central finding, the claim that we'll go through here, is that when subjects, human subjects, are making free volitional decisions to press a button or not press a button according to their own schedule, just internally, according to their own desires to press the button or not. The claim is that brain activity precedes the conscious awareness, the subject's own conscious awareness that they're about to press the button. And this sort of thing, this is the general experimental paradigm. And I took this from Patrick Haggard, a nice review in Nature Reviews Neuroscience from Patrick Haggard, and I recommend this review for any of you who get curious and want to look at this further. This is the basic diagram of Libet's procedure. So a subject sits, it's very simple. I mean, this is startlingly simple. A subject sits in front of this little button box, and there's a timer on a clock face going around, and it's making a complete revolution every 256, every 2,500 milliseconds, every 2.5 seconds. It's making a complete revolution. And the subject is instructed just whenever they feel like it, whenever they have the urge, whenever they feel like it, to reach out and press this button. And when they reach out and press this button, simply to note visually where the hand is pointing on the clock. So the time of their first urge to press the button, they're supposed to note that. And then they go ahead and complete the button press. They note the time of the urge, they go ahead and complete the button press, and then the data that the experimenters record is simply the clock hand was at position 4. Okay, so it's a subjective estimate, the subject's subjective estimate of when they first felt the urge to press the button. And of course, the actual button press occurs a few hundred milliseconds after that. Now, at the same time the subject's doing this, they have the EEG leads on their head. There's usually some EMGs and the EMG electromyograms to actually measure muscle contractions. And here's the basic observation. This is Libet's result right here. So the observation is that you time movement onset. So it's easy for computers to time when the subject actually starts moving, either through an EMG electrode or through the time that he actually presses the button. And then the subject is reporting what time he first felt the urge to move, and the observation is, and this is typical, that urge to move occurs 300 or 400 milliseconds before you can actually measure the onset of movement. But the uncomfortable observation, the really shocking thing that Libet found, is that if you look at the activity in premotor cortex and supplementary motor area, what you see is that there's electrical activity that starts rising at least several hundred milliseconds before the subject is consciously aware of an urge to move, and sometimes up to 1 second before. So there is brain activity. The message is that there is brain activity that has, in essence, already set this process in motion. This is Libet's fundamental conclusion, and something that's been confirmed by several laboratories over the ensuing decades. And the notion is that somewhere the brain itself, these freely guided movements, the brain itself is planning this, and consciousness only rides along on top of it. You only become consciously aware a few hundred milliseconds later of things that are already going on in your brain. And this has been the subject of intense controversy over the 3 decades since it was first reported. There are all kinds of, from the point of view of experimental psychology, just measuring human behavior, there are all kinds of problems with this thing. So one argument might be, well, what the subject's really doing is directing their attention to the clock. So they've got to be attending to the clock and watching that clock thing go around before they actually feel an urge to move. And what we're seeing right here is simply a redirection of attention to the clock face before an actual urge or decision to move has come on. So maybe this is related to attention. Another problem is that the subject has to remember this, right, until after he's pressed the button, and then they're giving reports to the experimenter. So there's some memory involved, okay? What time and how did exactly you register the time on the clock with your internal experience? So there are lots of problematic things with this experiment, and people haven't known quite what to make of this over the ensuing decades. But it's something that has been really important because it is the source of a lot of the things that we hear even today about how little our conscious control has to do with our behavior and how much things that are preconscious or subconscious determine behavior, and then perhaps our conscious labels about I did that or I chose to do that get laid down after the brain has already been active. So that's the central claim. Is that clear? So for anybody who walked in here not knowing who Libet was or why he's been important. By the way, this is the most famous neuroscience experiment ever among philosophers. Philosophers and to some extent psychologists, but certainly among philosophers, this and the discovery of mirror neurons by Rizzolatti. And if you go to an average medical school and ask an audience this size, how many of you have ever heard of Benjamin Libet? It's about a quarter of them that will raise their hands, 20–25%. But in philosophy circles, this is the most famous neuroscience experiment ever. Okay, so just for those of you who are not brain cognoscenti, I just wanted to show you, this is the motor area, the classic motor cortex, just in front of the central sulcus that we normally think of actually being involved in execution of movement. But in front of motor cortex are this premotor area right here called PMD, and then there's the supplementary motor area and the pre-SMA. And it's these areas up here that have been implicated due to numerous kinds of observations now in motor planning of events long before you execute them. And it's these areas that are thought to give rise to this readiness potential, this thing in the EEG that Libet discovered. Okay, so I want to skip to the paper that I think, in my opinion, overcomes some of these methodological problems with the measurement of behavior. And so this is a paper from Matsuhashi and Mark Hallett at the NIH, published in the European Journal of Neuroscience. I'm glad to be able to report that, in 2008. And Hallett and Matsuhashi wanted to develop a paradigm where they could get away with some of these subjective problems about reporting volition. This business about attention, about memory, we don't want all that stuff complicating our paradigm. Somehow we want to make a measurement of when the subject actually starts consciously planning the movement, but we don't want to have to rely on some of these loose methods that people have relied on in the past. So they developed something called the veto task, which I'll describe to you. And the aim is to remove the subjective aspects of the verbal time reports and provide objective measurements of the time of the intention to move. All right, so I'll describe the procedure to you, and you can determine for yourself whether you think they've succeeded. So first of all, the subjects are, as in Libet's experiments, the subjects are asked to perform sequences of freely initiated finger movements at intervals of 5 to 10 seconds. So they're discouraged from any kind of precise timing, any kind of regularity. They're instructed, press the button or move the finger when you feel the urge to, but try to space them out a bit. You can have them separated by 1 or 2 seconds or 10 or 20 seconds, but just space them out a bit. On average, try to hit for something about once every 5 to 10 seconds. And this is a quote from the paper. They were instructed to make brisk movements as soon as the thought of movement came to their mind, and not to count, not to keep time, and not to think about the movement during the waiting interval. The waiting interval is the waiting interval between urges. So now, here's the key thing. Simultaneously, while these subjects are making these sequences of self-timed movements, the experimenters are presenting little auditory tone bursts. Beep! Little auditory tone bursts at pseudo-random intervals of 3 to 20 seconds, all right? And the subjects then are instructed to ignore the tone if it occurred outside the brief interval between movement intention and execution. But if the tone occurred after the subject decided to move, the subject should cancel the planned movement if possible. Now let me explain that. It's well known in experimental psychology that you can get a signal to move, and I can signal it to you on a computer screen, but if I give you a veto command within a few hundred milliseconds after the signal, you can actually withhold it. Even though your instructions are to move as soon as possible after you get the instruction, after you get the signal, if a veto signal comes within a reasonable amount of time, you can withhold that movement and avoid making it. So this is what these subjects are asked to do. This is nothing terribly novel or crazy. It's well known that subjects can do this. So this is a veto or a cancellation program. Now remember, these tones are coming along randomly. And while the subject's doing this, of course, we're going to measure the EEGs and we're going to measure the electromyogram from the hand throughout the experiment. And notice here that there's no oral reports. They're just trying to keep things simple here. There's no oral reports whatsoever. So the subject's just sitting there, just when he gets the urge, doing what I'm doing right now, all right? But these tones come along at random intervals, right? So this is a random string of tones. And if the tone comes after I get the urge, then I withhold that movement. Okay, that's all they're doing. They're not reporting, they're not looking at any clocks, they're not reporting their memory of any clock number or nothing like that. Just can you veto it or can you not? So that's what they're doing. Okay, so these kinds of, this is a kind of time series of events from one example trial. Now there's no real trials here, right? Because these are self-initiated movements. This is nothing the experimenter has control over. But here's an example where a tone occurred at this particular point in time, and it was followed some gap later. It doesn't matter exactly right now. We'll get quantitative in a minute, okay? But it's followed some gap later by the subject's actually executing movement. And this squiggly line right here is the electromyogram. It's an EMG recording from the hand. So you can see that the subject actually executed a nice hand movement right here. And so the time intervals that really matter here are the time before the tone. So the tone occurred right here, and there's time before a tone that we'll analyze and times after the tone that we'll analyze. And basically what we're going to do is just look and see what the interval is between the between the occurrence of a tone and the actual execution of the finger movement. And this is the kind of data that they get, and this is interesting. So what we're plotting on the vertical axis are tone events. So these are the timing of tones. Each little tick mark on this vertical axis is a tone. And you can see that there are quite a few tones occurring out here. And then we get this dip right here. And time 0 over here is the time that the subject actually executed the movement. This big black line here is the time the subject executed the movement. So what they're doing here, they're taking 200 or 300 movements from the person, and they're simply aligning all of these traces to the time of the movement from each of 300 movements, and then they're plotting the time of tones that occurred before and after. And because tones are coming in a random sequence, if the subject could not do this experiment, if the subject were just ignoring the tones, you would expect a constant level of tone events across time here. But in fact, the subject is successfully vetoing in a certain interval, and so what you see is that in this particular interval of time here, just before the movement, there are far fewer tones observed. And that's because the subject's vetoing tones that occur in that interval. And so you never get an actual finger movement, and therefore you don't see tones in that interval of time before the movement. Is that clear? This is critical, okay? So then what they do is simply measure this time, the time at which the tone events depart from their regular random sequence and start becoming rarer and rarer as you approach the movement event, the measured movement events. And you can see that this is a nice way to sort of quantify this veto period. And once you get here, once you get to this line right here, you're within a certain time period of the actual movement. And if the veto signal comes that late, if the tone occurs here, you can't veto it. You reach what they call the point of no return. So this is actually nice evidence that the subjects are able to do this veto task, and furthermore, it provides a quantitative metric for the timing of this interval, this conscious intention or plan to move, because you can fit a nice sigmoid to these data like this, and you can put a line down in the middle of the sigmoid if you want to, and declare that the time at which the intention to move was consciously formed, and after which you're able to veto those things. So this is nice in the sense that you're not relying on subjective reports here. You're just analyzing the time relationship between the tone events and the actual movement events, okay? And in this particular subject, in this experiment, this turned out to be about 1.7 seconds this interval between the putative formation of the intention and the actual execution, 1.769 seconds. They analyzed all of the data in two different ways. Here they fit this sigmoid, and by the way, they can also fit a sigmoid to the other end to actually get a time for this point of no return. Here they just ran a smoothing kernel over these data, the tone bursts, and did a different calculation on the smoothing kernel. But you can see that this time, these are the same data, by the way. It's just different analyses, two different analysis methods. And the times agree reasonably closely, or about 1.76 seconds and 1.9 seconds. So, at the same time then, they're measuring the EEGs, right, these readiness potentials. And this is an example of an EEG measured in microvolts, this readiness potential over the frontal motor cortex. And it's bubbling along here at, you know, some rate, some noisy rate around baseline. And then suddenly out here at about 2.5 seconds before, it starts departing from the baseline. And there's actually a second, more steeply rising phase here. This is, they typically call this the early readiness potential, and this is the late. And so what Hallett and now Matsubishi did was to fit lines to these things to get an objective measurement. So they first fit a line to the baseline, that's the line of slope 0 to the baseline, and then they fit a linear regression line to this early potential, and then they fit a second regression line to this late potential. And where these regression lines cross, okay, these crossing points are what they call, this is the onset of the late potential, and this line out here where it crosses is the onset of the early potential. And these are the data then that they measure. So they lay a line down here to measure the onset of the volition to move the hand, and they measure these times for the onset of the potentials. Now we can quibble about some things about the procedure here, but basically I feel like it's pretty good. And here's the data that they get. And the key comparison here is between BP1. BP is the readiness potential. That's a long German word that I can't pronounce. Some of you probably know what it is. But this is the early readiness potential, and on average across seconds, now across subjects, these are all average data. On average across subjects, this readiness potential here is leading the actual T, the timing of the intention, you know, this measured timing of the intention to make the movement. It's leading it by about a few hundred milliseconds, as typically happens in Libetian studies. These things down here are interesting. The second phase of the readiness potential is actually following this conscious intention, presumed conscious intention to move. And of course, the point of no return is on the order of 100, 200 milliseconds, and that's typical in these kinds of experiments. But this is the key result right here, and that is not a terribly strong result. These are first and third quartiles on measurement. The dark bars here are the median measurements. These are 25 and 75% quartiles, and these are the extent of the data. And this is not a big result. This is 15 subjects. It's significant at the 0.023 level, but to my mind, this is the best, most quantitative attempt to replicate Libet in a objective manner. And they get the result, but I would say it's a weak result. There are actually 4 of the subjects. They provide the subjects for all of the data in the paper, and for 4 of the subjects, average time of T actually precedes the readiness potential. So this is not something that holds in every subject. Stuart.
Just a technical point.
Yes.
Is the EEG averaged over many trials?
Yes. Yes. These EEG signals are too noisy to do single trial experiments. The signals are too noisy, and the effect is too small. So yes, all of this is representing average data. Okay. So look, what are we to make of this? I mean, it's a moderately weak effect. I mean, it's real, I believe, but it's moderately weak. There are some technical issues that I have with this. One of my technical issues is that they actually lay this line down and they measure the onset of intention to move. They put it down in the middle of the sigmoid. Well, you know, why wouldn't you put it down at the beginning of the sigmoid where it starts to deflect? And if you put it down at the beginning, then you move the T measurement, you move this thing a couple hundred milliseconds further into the future. And if you did that, this effect here would likely disappear completely. So I'm not completely sold necessarily by these data, but Hallett and many other people claim to have replicated Libet's fundamental experiments. Okay, so about 5 years ago, a student at Stanford, a graduate student, came to me and said, well, it just came up in conversation. He had an experiment, a Libet-style experiment that he thought should be done because he was worried that some of the effects that are being reported in the literature might be an artifact, a signal processing artifact, or at least if not an artifact, that could be a little too strong, but if you looked at the data in a different way, you might come to very different conclusions about what's going on. And this guy is Matt Kaufman, and what Matt's basic notion is the following. That what's really going on in the brain is that there is an underlying time-varying, somewhat random signal. So somewhere in the premotor cortex or somewhere in the brain, there's a time-varying random signal, and you can think of that signal as something related to the desire to move, maybe just a general desire to get up out of my chair, or to move my hand, or to scratch my head, or whatever. We don't know it's always dangerous to put psychological labels onto electrical signals, okay? But people do it all the time. But you can think of this as a proclivity toward movement, maybe not an active desire, but maybe nearness to threshold for executing movement, or you could just think of it as any underlying biasing, random biasing signal. So what if there's just this randomly varying signal going along in the brain at all times that corresponds loosely to some kind of tendency to be near threshold for executing a movement? Well, the idea is that maybe what we're doing volitionally when we're in an experiment like this and just generating at random times finger movements whenever you feel the urge, maybe what we're doing actually volitionally is setting a threshold, some sort of threshold on this underlying random signal. And when that signal crosses some threshold, then we'll go ahead and execute the finger movement. And as long as that signal's hovering down around under threshold, then we won't execute a finger movement, okay? And the notion is that the actual event that corresponds to the conscious intention to move is this time right here where the signal crosses this threshold. And here's the suspected, what we in our gut suspect is a methodological flaw in these studies so far. The problem is that if you take a whole bunch of trials where you have had a positive choice to move, and if this kind of picture is really true, that this is a result of a random fluctuation above some kind of threshold, and you take a bunch of these trials and you average them together, then inevitably you are going to see a rising potential before the actual hand movement just because of autocorrelation in this randomly varying signal. Okay? And that's the gut feeling here. And what you would really like to know is what happens on circumstances where you decide not to move. What is that decision, what does that signal look like when you're not moving? And of course, all the experiments I've described to you, Libet and Hallett, it's all based on the decision to move, never on a decision not to move. So here's the scheme. The scheme is that, one trial, this random signal crosses this threshold at this point in time. On a different trial, it crosses at a somewhat later point in time. On a third individual trial, this random signal may cross earlier in time. So these three arrows that indicate the crossings here are all happening at different points in time. But if you take 100 or 200 or 300 of these things and average them, what you're going to get is some signal that looks like that. And that's inevitable. That has to be the case because of these autocorrelations in the signals. And so you might conclude that there is some potential out here that the brain is in fact already deciding to move long before you have experienced an intention to move, but this might just be a signal processing. I don't know if we want to call it artifact or not. It might just be a result of the signal processing. And so Matt said, Bill, we should do this experiment. And I don't have EEG stuff in my laboratory, but I think it's a great experiment. So what Matt wants to do, his new feature is that he wants to capture neural events that precede no-go as well as go decisions to test this hypothesis of this underlying random variable that's going along. Now, Matt's design involves a price, and a lot of Libetian people would say it's too steep a price to pay. And the price is that we give up temporal spontaneity even though free choice remains. All right, what do I mean by that? Here's what I mean by that. The subject is going to be sitting there listening to these noise tones, and we're going to be playing these noise bursts, boop, at random intervals, and in this sense, this experiment is just like Matsuhashi and Hallett. So they're going to come at random times between 1 and 15 seconds with a flat hazard function, so that the subject really cannot anticipate and cannot guess when the next tone is going to occur. And then when each tone occurs, and not before each tone occurs, and this is critical, you depend on some cooperation from your subject here, the subject immediately decides whether to press a button or to withhold a response. Okay, so this is critical. This is what Matt is grabbing. He proposes to grab the withholding of a response as well as the actual committing a response, actually making a response. Now notice what he's giving up. What he's giving up is that the subjects come under temporal control of the experimenter. So in Libet studies, the subjects are just sitting there making the finger movement whenever they want to, whenever they get the urge to. But here, what's happening is that Matt wants to seize control of that in time and present the tone, and then have the person decide spontaneously, of their own free will, their own volition, whether to make the finger movement or not. So this is critical here. Subjects are instructed not to establish a pattern and not to choose one alternative exclusively. So you can imagine some subjects just going commit, withhold, execute, withhold, alternating. That defeats the whole purpose of the experiment. Any kind of strategy like that, any kind of cognitive strategy like that, defeats the purpose of experiment. And of course, if they just were to choose one alternative exclusively, the experiment's out the window. So you would require some assistance here from the subject. Of course, we're going to measure the EEG and EMG throughout. And the nice thing, like Hallett, there are no oral reports here. The subject's just listening for the tones, and then when they hear the tone, at that moment in time, they decide, do I make the movement or do I not make the movement? So now we have two kinds of trials, right? We have ones where they're executing the movement and ones where they are withholding it. And here's the prediction. The prediction is on a go trial now, of course, when you press the tone, if the subject decides to go, we're going to predict that this randomly varying signal is above some threshold like this. But on other trials, where the subject decides to withhold the experiment, we're going to predict that the randomly varying potential is below this thing. Now, of course, in principle, you could adjust the relative number of go and no-go signals just by volitionally saying I'm going to have a higher threshold or a lower threshold, right? But the key thing here, the key point here is that we get both kinds of experiments. And the prediction is, if we have this kind of randomly varying signal, and you're just going in and polling that randomly varying signal to generate the decision to commit or not commit, the prediction is now that we should see these rising potentials long before the tone on go trials, but we should see negatively going potentials long before the tone on no-go trials. And this kind of thing has never been tested. It's never been incorporated in any aspects of this experimental design, to my knowledge. And to my mind, this would give a different kind of interpretation to these experimental results if this were done. So just to try to bring that out more in a crystal clear fashion, here are the predictions from Matt's model, and here's the predictions from the traditional Libet model as it's been interpreted by many people since then. And some of you will know Emil, who I'm talking about here. So the traditional notion coming out of Libet's experiments is that the brain is sitting there in some kind of quiet state on single trials. The brain is sitting there in some kind of quiet state, and then suddenly the brain, somewhere subconscious, the brain starts deciding to move. And we see that happening, and the brain starts deciding to move, and we get this go potential, with a movement here. Whereas on no-go trials, you simply see a continuation of this baseline like this, and there's no decision to move, right? But on Matt's model, what we should see is this randomly varying potential, and whether the subject decides to go or not go should be dependent on where the potential is just before the tone, at the time of the tone. And if this turned out to be the case, and we don't know it because the experiment hasn't been done yet, but if it turned out to be the case, then I think that would put a different light on these kinds of measurements. this randomly varying potential would still contribute, okay? It's real and it would still contribute, but the conscious act to decide whether to make or withhold the movement would really be a matter of some part of the brain going in here and polling this signal at some point in time in order to reach the outcome. And this long extension of the signal backward in time then just becomes a matter of autocorrelation in the neural electrical activities statistics. Okay, so that experiment hasn't been done. I've posed this now to 3 people who do this kind of work. I've had no takers yet, but I intend to keep trying unless somebody convinces me that this is a bad idea. But what's at stake here? So I've taken you through some of the experimental data. So let's zoom back out here and say, is this even an important question at all? What's at stake in here? And there are fundamentally, to Libet's original question, his original experiments, there are 3 possible outcomes, right? So one possible outcome is if you wind up doing the experiment properly and avoiding all artifacts and being nice and quantitative about everything, that conscious awareness, I'm going to call this CA, conscious awareness occurs before the measurable neural events. And that would be a very weird result from a neurobiologist's point of view. This would be Descartes, right? This would be Cartesian's mind out there in space somewhere giving commands to the brain. And to a neuroscientist, that seems unlikely. Not impossible, perhaps, but it seems unlikely to a neuroscientist. And if I got this result in my laboratory, I would just suspect that we weren't measuring the correct neural signals yet. You know, that this would not convince me that there is a non-embodied mind in some sense interacting with the brain. Here's another of the 3 possible outcomes, that conscious awareness occurs simultaneously with the relevant neural events. If we ever get our finger on the relevant neural events, that these 2 things occur simultaneously. And I think that would be okay. I mean, that's like, you know, sort of changing the voltage on a capacitor, measuring capacitive current and saying, you know, which happens first, the change in voltage or the change in current? Well, you know, it's not. One doesn't happen before the other. There is no delay in the C in the little equation for capacitive current. And it could be that conscious awareness and the neural activity of the relevant events are 2 sides of the same coin. And I think that's still in play for the reasons that I've told you. I don't think that's been excluded yet. It may be excluded over the next few years, but I don't think it's excluded yet, Libet and decades of work notwithstanding subsequently. Third thing possible is that Libet's right, that the conscious awareness happens after the neural events, and that Libet's right. And this would change, we'd have to start changing, and some people are already changing the way they start thinking about conscious awareness and its relation to our behavior. We have this intrinsic feeling that our conscious intention is what causes us to reach out and move the finger. But the message here would be that it's other events going on in the brain that actually cause you to reach out and move your finger, and you only become conscious of it at some later point in time as that signal gets sufficiently strong to cross a perceptual threshold. So I want to make 1 or 2 comments about that, and then we'll stop and have a discussion. In a sense, this outcome here, if it's correct, and of course this is what Libet and many other people following him argue, if this is correct, it's not shocking. We know that there are many signals present in our brain, and they influence our behavior without our being aware of them. So these are things like innate biases, like priming effects, and such as this, and they are present in our brain, and they influence our choices. Social psychologists love to show us this kind of thing, right? That we think we're doing something for one reason, whereas in reality, they're shaping our choices by presenting us with priming materials that we're not even aware of. And the most spectacular example of this I have ever seen, I mean, this is the most impressive scientific paper I've read in the last 2 years, is a paper published by an Israeli group entitled, a very calm title, Extraneous Factors in Judicial Decisions. And you know, we all know that there are extraneous factors in judicial decisions, but these Israelis studied Israeli courts where prisoners are appealing for release on parole. And in Israel, these judgments are made by magistrates who sit there and do these lickety-split, 1 every 5 minutes, and they do it all day long for 8 or 9 hours, and they have 2 breaks for food in between. And these guys went out and accumulated a bunch of data from very specific judges and they plotted the proportion of decisions that are favorable toward the prisoner as a function of the time during the day. And the dotted lines right here are the food breaks. And look at the numbers on this axis. It goes from 65% in favor of the prisoners at the top of the day to 0% just before that first food break. And then it bounces back up to 65%, and then it's down to 10%, bounces back up and down again. We might as well be flipping coins. This is true, right? I mean, this is outrageous. What are we doing here? And you could say, well, there's all kinds of things that might be covariant. Maybe they list the easy cases first and go to the hardened criminals later in a session. But there are all these controls, you know, where they plot the gravity of the offense, again, on the same axis, the previous incarcerations on the same axis, the number of months served in the term on the same axis, and the proportion that have participated in rehab programs, and they're all flat. They don't account for this. Stuart?
Someone should do this with professors grading papers.
Exactly. When I saw these data, I had just come from reviewing grant applications. In a panic, I rolled out the record to see whether my choices showed any pattern before lunch break or after. And fortunately, they didn't. And I think it's because I have more knowledge of this field and grants, and my prejudices are based on other things. You know, like what I know about the candidates. But that was exactly my thought, Stuart. Okay, so those things are going to be in the brain, right? There's no doubt about it. Another thing that I should tell you about is that there's corroborating evidence for this from electrical stimulation of the brain. And I'll just tell you this very quickly. If any of you haven't seen this paper, this is an unbelievably cool paper. So this is some neurosurgeons, and when they're doing neurosurgery on people for epilepsy, intractable epilepsy, they have people inside surgery conscious, fully awake and conscious for purposes of the clinical treatment. But they take time, those neurosurgeons take time to play while they're in there. And they stimulate the brain. And what they did, they accumulated a bunch of stimulation in premotor cortex, and then a lot of stimulation sites across many patients back here in parietal cortex. Now what they found is that if they stimulated premotor cortex, electrically stimulated, frequently they would get subjects to move their arms or move their legs. And you ask the subject, why is your leg moving? And the subject says, I don't know, I didn't do it. You must have done it. Okay? So the subject is aware that their limbs have moved, but the subject disclaims responsibility for that. Okay? But when you stimulate back here in parietal cortex, the subjects will report that they feel an urge to move. They want to move their arm, or they want to move their leg. And sometimes they will even claim that they did move their leg and did move their arm, even though objective measurements of the EMG show that it didn't. Okay? So there's ample evidence that electrical stimulation, electrical activity of the brain alone can be responsible for this subjective urge to move. All right? So these are things that we have to take into consideration when we're talking about this. And we're all okay, I think, with some biases in our behavior, but it's a bit more disturbing when even the most straightforward sense of conscious volitional control, like simply deciding to move a finger, falls into this category. So what do we think about this in the end? Well, this is a quote from a colleague of mine, Dan Schachter, who's a memory psychologist at Harvard, actually, at a meeting. And he said something to me in casual conversation. This is 15 years ago, and I still remember it. So it made a big impression on me. And what Dan said to me, I said, what do you think of this Libet stuff? You know, is our consciousness just following along behind brain events and all of the stuff we think we have conscious control over, we really don't? And what Dan said to me is, he said, well, I don't mind showing up a bit late as long as I get to play once I arrive. Now, what he meant by that is that we may not have control over the moment that something enters consciousness, but when it does enter consciousness, the fact that it enters consciousness has an effect on the sequelae. So, for example, if we took those data from the Israeli judges and we showed it, here's how you're behaving, and they became consciously aware of the bias that they have, they could, in principle, change their behavior. But until you become consciously aware of biases, whether they're racial biases, whether they're national biases, whether they're hunger biases, whatever, until you become consciously aware of them, there's not a damn thing you can do about it. Okay? And it's possible then, I would say, that consciousness, conscious awareness, may be a part of the causal story of our behavior, even if it's not the initiator in many cases. And that's as far as I've gotten in my thinking, and I will stop now and happily entertain a few questions. Thank you very much.
Thanks, Bill. Well, we have plenty of time. Let's have another comment.
Bill, supposing you do this, supposing you get the temporal variation, not by asking the subject to do something really undefined, like press the button when you think about it, but you give them a little task which varies slightly from interval to interval, and they're to press the button as soon as they finish the task. I mean, a little bit of mental arithmetic would be an obvious example. Then I don't know whether you still get a readiness potential.
I don't know either.
But you would have all the other conditions of a Libet experiment if you did that.
Except I don't know if you'd have a thing you could hang the no-go experiments on. So I mean, that's the real flaw in all of these things, in my opinion, is that we don't have a negative control. We don't know what the other side of that equation looks like. So we can't see if there's a constant baseline and we actually get a departure from the baseline that initiates or precedes movement, or whether we have this random
As Edmund Rolls once told me, you don't need it controlled if you get no effect.
That's true. That's true. That's true. If there's nothing to control for, yes, I agree.
I think this,
Can you speak up, Lance?
there is a temporal spontaneity in this decision making.
Yeah.
Even in mechanics, like Post-Libet, there is always a continuity. There is no jump from static state to dynamic stage.
Okay.
So in this Libetian experiment, it is so obvious that because they are all instructed to do things.
Yeah.
So you never know what's happening in the brain because they know what they have to do.
Yeah.
I can't imagine a spontaneous brain stimulation that leads to an action. There could be a continuous,
Yeah.
So could you repeat
I think the question, if I understand you correctly, that behavior is intrinsically dynamic, and brain activity is intrinsically dynamic, and the whole Libetian program is mistaken from the beginning in thinking that what you really have is some kind of flat baseline, and then spontaneously some event happens, and that's just a fundamental mistake and wrong-headed to begin with. And my sympathies lie with you on this. And I would like to have experiments that actually test that. And I'm trying to think of ways to do it. But I don't know if I've come up with a compelling, what did you think, if I can ask you a question, what do you guys think of the experimental design that I proposed?
Very interesting. Only this, the conclusion that then you say this most likely that Libetian experiment. That I follow with that.
Okay, so I'm
from the philosophy business, obviously is Cartesian. I don't know.
I'm somewhat sympathetic to you. The reason I say that that's perhaps most likely is because we know on other grounds, we know that there are all of these unconscious biases to our behavior, right? So even if there is some random variable that we're going in and sampling at certain times to decide to make the movement or withhold the movement, that random variable may have big offsets on it. Maybe the experimenter has just encouraged me to increase the frequency of movements. So I'm gonna lower my threshold for making a movement. Or maybe the experimenter says, you're making too many of them, try to just tone it down a little bit. And it's going to be a bias signal on my criterion, basically. I'm very sympathetic to you, but we do know that there are lots and lots of biases and different ways of controlling biases. So I want to be as open about this as possible. But I do think the field has jumped the gun on this.
I just want to add something. There are other inputs that may add to the causation. Because you have to see things watching the floor,
yes,
and the thing, and
yes, or hear the tone.
Exactly. So without the medium of mind, I don't understand how you.
Yeah.
There's two questions next door, so perhaps we'll take you in turn.
So the assumption of a random variable then,
you could also start to gauge that from looking at the frequency statistics. So do you get something like a Poisson distribution, or if they're judged to say don't move more than once in every 10 seconds, do you get more of a uniform distribution?
That's a great question. So that's a great question.
Can you just repeat?
So the question, repeat for the microphone. Question is, if we really believe there's a random underlying variable, and what we're going down, we're just following this variable, and if it's above some threshold, we make the movement. If it's below, we don't. Then the times of occurrence of the movement should follow some very precise Poisson statistics, if it's truly random. And has anyone even looked at that? And the answer is that that's a more sophisticated analysis than most people in this field do. But a very nice paper just appeared last year in 2011 from Itzhak Fried's group in UCLA. And they actually do show exponential fits to the times of movements that are consistent with the subject not having any pattern. Now this is tough to do, right? If somebody tells you make random movements, it's very hard for humans to make random movements. We always insert patterns into our movements. So it's actually quite surprising how exponential those work. But that's the only paper I've seen that tests it that way.
And your neighbor?
I was wondering the experiment you were proposing, the one where...
My imagination.
Yeah, it's just where there was a graph at the bottom of the slide where the person will decide not to do the movement.
Yes.
For some reason, I'm assuming the y-axis is just the voltage on the EEG.
That's right. That's right.
The voltage drops.
That's right.
I was wondering why would it drop? I mean, the person is still going through a thought process, and the neuron still being active, just going through a different state.
Now the notion is, and I'm sorry if I wasn't clear about it, the notion is that there's this randomly varying signal in the brain, and when you hear the tone in the proposed experiment, when you hear the tone, you go in and sample that variable. And if that variable is above some level, you decide, I'm going to press the button. But if that variable is below some level, you decide, I'm going to withhold my response. Now, these voltages in the brain do not change instantaneously from millisecond to millisecond. If it's down at a low thing at 1 millisecond, it's likely to be low at 10 milliseconds from now. That's what I meant by their autocorrelations in these things. The time courses, the time filters on these signals that are actually in the brain circuits, not in electronic circuits. Those time filters are finite. So what I'm just saying is that when you get something where he decides not to go, I predict that the voltage is down, it's below the criterion, and furthermore, it will have been down for some few hundred milliseconds just because of these autocorrelations in the signal. Does that make sense?
Yeah, I was wondering why that prediction? Why the prediction that it has to be low? That's simply a model, and the model may be wrong. That would be a model that we want to test. But we predict it's low because it seems natural that there's some varying signal. And when that signal is high, you decide to move, and when that signal is low, you decide not to move. It could be wrong, but that's the hypothesis.
Can I ask a question about, then we'll come back to it,
Okay.
about somebody who's really trained well to respond to stimuli very quickly, and I was thinking of Formula One Grand Prix drivers.
Yes.
Lewis Hamilton is our favourite. You know, your delay of 2 seconds seems you'd have crashed into the barrier and died.
He did, yes.
Yeah. So somebody ought to wire up Lewis Hamilton, I think. They've got everything else wired up in his car. but is it different if you have people who actually spent their life training themselves to respond to stimuli very rapidly?
It's a great question, and almost certainly it's different. Professional athletes are different from those of us who are normal human beings. And this has actually been shown in measurements of baseball players, and I presume cricket players, of their eye movements and how rapidly they move their eyes in response to a moving stimulus. They are on the outer end of the distribution. And this also goes back to your point about behavior is dynamic. We're not planning pieces of behavior one at a time. In a Grand Prix racing car, or even something as simple as taking a pot out of a cabinet and putting it on a stove, that's a continuous sequence of muscle contractions that follow dynamically one after the other and are very frequently habitually ingrained. And there may be some stimulus that sets that off, like I want tea or I want to boil an egg. But that thing is set off dynamically, proceeds dynamically with some sensory feedback and guidance, but not totally dependent on it. So I think you're right.
It's interesting, and this is just a follow-up. Sorry,
Yeah.
As a follow-up, now in sports, there's some coaches who are saying, don't decide, go with your gut. So in fast-moving situations in football and baseball, perhaps even in Formula One racing, the coach's advice is don't make it conscious. React, and you're probably going to be more accurate. Now, what that says about conscious awareness and motor decision making.
You seem to say that you needed more data, that is more experiments.
Yes.
How many more?
Well, you know how it is in experimental science. If you do a really good experiment, it generally raises some more questions. So I can't tell you that how many more. But I do think, you know, different levels and sequence of experiments, you get new and different insights, and you become more convinced of one thing versus another thing. But I can't tell you how many more.
Bill, what another way of putting the conundrum is, Libet made strong assumptions about what the correlation of making a decision was. Okay. And he also made an implicit assumption about what the correlation, if any, of the rise of the readiness potential is. So one thing you could ask is, if you did very subtle experiments, is there something the subject can tell you about the early phase of the readiness potential? Suppose you look to see whether subjects could say whether the readiness potential was rising more rapidly or less rapidly in different blocks of trials. There might be a conscious correlate of it, but it's, you know, maybe just feeling jumpy about, you know, the fingers feeling itchy or something like that.
Yeah, and Stuart, I've wondered, I mean, there are all kinds of permutations about the subject and its participation. I've wondered if I were in the experiment, could I defeat this standard readiness potential measurement just by sitting there thinking left, 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 but I moved right.
It's not Libet that discovered this, of course.
Sorry?
It's not Libet that discovered all this.
You know the history better than I do. Why don't you tell everyone?
I think it was a German neurologist called Kornhuber.
Yeah, who discovered the readiness potential.
Yeah, but I mean, everybody knew that it was moving before you obviously made the decision. Libet's only a sort of icing on the cake to me. People just didn't worry about it.
So Libet's only contribution was to get us all to worry about it?
No, I agree. They should make a crass assumption about what it means.
Yeah.
I think that's a good note to end on. Thank you very much.
Okay.